Yeehaw class. Since I'm not in today, I thought we would try something a little different than usual and do a zaption of yesterday's notes. So in some classes we got through the entire set, in a couple we didn't. So either way, we need to check your comprehension on this stuff. So I thought a zaption would be a fun way to do that. So if you look on the first slide, I've got again a hydra. So the theme with the hydra is if you cut off a head, two will grow back. And so we had Hercules here, and he was fighting the Hydra. And the theme would be that he would cut off a head, and two would grow back, and he would be like, why? And so we would go down here to the recap. And just to refresh your memory, DNA has four different nucleotides in there: A, G, T, and C. What you need to know is that DNA is a double helix. Remember that DNA is basically just those four letters in a random sequence. And that since DNA is a double helix, it means there's two partnering strands. So if we remember our skits and projects, that's the reason we had a couple being split up, forming two new couples. That's because that's what DNA does when it replicates. If you look down here, we have a little reminder that you guys are pretty comfortable with, I know, but just to refresh your memory and ingrain this into your heads, G always goes with C, and A always goes with T. And if you can look down into the bottom picture, you can see that A is always with the T, and G is always with the C. And if you look on top of the two sets of strands, so remember I've got two partnering strands. So I've got one right here and the other right there. And above it, we've got an arrow going that way, and then we've got an arrow going that way. And what does that mean? It means that the DNA is once again anti-parallel. So one strand points to the right, the other's going to the left, and that's why we have the A's pair up with the T's and the G's with the C's, and vice versa. This is another lovely picture of DNA. So right here, we have the sugar phosphate backbone. It forms the frame of the DNA spiral staircase. And then around, or the steps, I should say, on the staircase, are the nucleotide base pairs. So I've got G going with C in this circle, and then I've got A going with T in this circle, T being the blue, A being the yellow. And G would be the red, and C would be the green. Pretty straightforward. The metaphor for DNA replication is if I have a candle and it's lit, and I got another guy over here who's got a candle and his is not lit. What would happen if I try to light his candle? Well, I light his candle and he gets a flame, but in giving him a flame, mine doesn't go out. So the important thing is just because I replicate doesn't mean I lose my DNA. The main problem we got to solve is that in order for that to happen, DNA has to copy itself before the cell divides. Don't worry about the synthesis phase or the S phase. You don't really need to know that. What you need to focus on this slide is that DNA has to copy before the cell divides, before it splits. And it needs an identical copy before the cytoplasm can split. So to refresh our memory, these are the same. They're one chromosome. This is one chromosome, and this is one chromosome. The only difference is that this one, bottom, has two copies. So I got a copy there, and I got a copy there. Now, if it was a different copy, one of these guys would be a different color or a different length. So why don't we do the top one being purple? So if the top chromosome would be purple, that would signify that it came from another parent. Or if it was shorter, it would be a different chromosome altogether. Remember that chromosome 1, your first chromosome is your longest, and if you have 23 pairs, chromosome 23 will be your shortest. And The chromosomes get shorter and shorter in between. You need to remember that you are made out of eukaryotic cells, which means that they have a true nucleus. The nucleus is there. And that DNA, since it's in the nucleus, the replication happens inside the nucleus. All right, tying this into our skit. Remember, RNA. So, medical detectives class, you guys have heard RNA. If you're not in it, don't worry about it. But RNA is basically DNA's kid brother. So it's got one strand, and it has a few different chemical properties. But overall, it's pretty similar to DNA. It has a variety of jobs, but in DNA replication, its job is to be the wingman, while DNA is waiting to replicate. And the three basic differences are RNA only has one strand. It has a 5-carbon sugar, a pentose, pentagon, pent is 5. It has a pentose, but it's a little bit different than DNA's. Also, it has a uracil, which means it will have a U instead of a T. Again, I've crossed out the T to remind you that RNA doesn't have it. And here we have the lovely Goose and Maverick. Look at that mustache. That is saucy. And then if we go down here, we got the dudes from Bad Boys 2, and then Buzz and Woody. RNA visual. So RNA has one strand. So let's highlight it in pink here. RNA has one strand. And then if we do let's do DNA, and let's have some fun and do purple. DNA has 2 copies. That's one copy. This is the other. So that is one prime difference between DNA and RNA. RNA only has one. Bear with me. RNA only has one. The other difference is that RNA has a U. If you look at the color, uracil is orange, whereas thymine is red. So again, uracil is orange, thymine is red. But if you look at all the other nucleotides, adenine is yellow, guanine is the same shade of green, and cytosine is the same shade of blue. Adenine is the same. Guanine is the same, cytosine is the same. Point is, adenine will bond with uracil like it does with thymine. So if we put an A2T in DNA, in RNA, it's going to be A2U. DNA is anti-parallel. You guys know what this is from your skits and projects. But again, if I have a car going north, like in this picture, and we're going south, that means we're still going side by side but traveling in opposite directions. Oops. DNA is anti-parallel. What that means chemically is that DNA has a 5-carbon sugar, a pentose, remember? So I've got carbon 1 there, carbon 2 there, carbon 3 there, 4, and 5. You'll see the 3 is down here, and the 5 is up here. But on the right side, it's switched. So the 5 is down here, and the 3 is up there. So if you're going from the 5 carbon to the 3 carbon, on the left you go down, and on the right you're going up. That's why you have C's paired with G's and A's with T's, and vice versa. So step 1 of DNA replication is that helicase, our date stealer, splits up DNA like this. So the arrow is pointing through the DNA nucleotides, and it's going to split up those bonds and cause the strands to separate. And the spot that DNA separates is called the replication fork. So right where it starts to separate like that and like that, that is called the replication fork. After we make the fork and we start splitting the DNA, we got pathetic DNA that's broken up and sad and miserable. So what do? DNA has to find its buddy RNA, who is the wingman. And RNA is it's in the form of a primer, so that means it's just a small piece of RNA. So if you look here, RNA is just that little guy right there and that little guy right there. It's not quite as big as the DNA, it's just a little teaser to keep the DNA happy while it's waiting for a new strand, a new date. What you need to take away from this slide is that DNA is the wingman that, or excuse me, RNA is the wingman that gives DNA someone to lean on and introduce the DNA to a new partnering strand. And step 3, our last step, is that DNA polymerase finds the RNA primer and makes a new DNA strand. So first we got to let's first find our original DNA strand. So let's highlight it in, let's do orange. So original strand. Then we had RNA primer help out after DNA was single and pathetic. So what we did was we find our RNA primer, this guy right here, the red dot structure. Okay, notice it's just a few nucleotides long, it's not very big. And then the DNA polymerase, the green guy, what he does is he finds the RNA primer and he says, hey, you're with a pretty high stranded DNA, let's replicate and make a new partner strand. And so that's what happens. So then you have this DNA polymerase go that way and make a new strand, which you see right there. So the difference between the top picture and the bottom picture is that the top one shows the polymerase binding to the DNA strand, the RNA primer, and then in the bottom one it shows it actually made a new DNA partner strand. So now DNA is happy, it's not lonely and miserable anymore, and it has a date. Laying down the new DNA. So remember the anti-parallel nature of DNA. So what that looks like is that helicase is gonna go that way because it's unzipping the DNA. And what happens is your top strand, so we'll call it complementary strand number 2 in this picture right here, it follows the helicase in that direction. But since DNA is anti-parallel, the one that's labeled complementary 1, this guy, is going to go that way. So this yellow DNA polymerase right there is going this way, whereas the top DNA polymerase right there is going to follow helicase and go the other direction. And then this is the whole picture together. So basic points, again, this slide's pretty, it's kind of complicated. The main things you need to focus on are this. We got helicase, the date stealer splits up DNA, and he goes that way and splits up the whole thing. After helicase does that, DNA is lonely and miserable, so what happens is RNA primer shows up to keep it company. And then DNA polymerase finds the RNA primer, so there's the DNA polymerase right there, and it starts to make the new strand. And it goes opposite direction since DNA is anti-parallel. So on the top, it's going to follow the helicase and go the same direction as the replication fork. And then on the bottom, it's going to go the other way. Punchline? You got to know that DNA is anti-parallel. Don't necessarily worry about the 3 to 5. You're going to see that in your high school biology class, but for now, you don't need to lose sleep over it. You just need to know that DNA is anti-parallel and that it replicates in opposite directions based on the strand. You got to know the differences between DNA and RNA. So those differences, I'm mainly concerned that you need to know that RNA has one strand and it has a U. So let's write one strand and it has uracil. Whereas DNA has two strands and it has a thymine, a T. The 3 steps of DNA replication are this. 1. DNA is split by helicase, the date stealer. 2. RNA primer, the wingman, couples with DNA and keeps it company while it's miserable and mopey. And then we have 3. DNA polymerase finds the RNA primer and lays down the new partnering DNA strand, and then you have replicated DNA. So, hope that was helpful, and go ahead and feel free to look over any parts of this video that might have been confusing, and we'll come with any questions on Monday's class, and we'll go over those that day. Have a good weekend.